CRISPR's new frontier, revolutionizing diagnostics and disease prevention. Now, with CRISPR's transformation into a robust gene editing tool, we find ourselves at the intersection of biology and technology, a place where ancient bacterial defense mechanisms have been harnessed to revolutionize modern science. Originally, these sequences, found in roughly half of sequenced bacterial genomes and nearly 90% of sequenced archaea, served as a microbial immune system. They archived viral DNA, defending against future attacks. The enzyme Cas9 acts as the molecular scissors, guided by these archived sequences to precisely target and snip DNA. This system, refined through evolution, is now reimagined as CRISPR-Cas9, a technology lauded with a Nobel Prize for its potential to edit genes in living organisms. From basic research to biotechnological innovations and medical treatments, CRISPR is carving pathways previously unimaginable. Yet, what truly captivates is how this ancient defense mechanism, once a mere survival tool, now stands as a beacon for future possibilities in treating genetic disorders and beyond. As we continue exploring CRISPR's potential, we uncover more than just a scientific breakthrough. We glimpse the future of genetic engineering. And yet, there remains an untouched frontier. What ethical boundaries will we encounter as we delve deeper into this genetic odyssey? By altering the guide RNA, a major leap that turned CRISPR-Cas9 into a precise gene editing tool. This groundbreaking discovery opened the door for precise, programmable gene editing, revolutionizing our ability to alter genetic material across a variety of organisms. Emmanuelle Charpentier and Jennifer Doudna took this concept further, refining the CRISPR-Cas9 system into a tool simple enough to be wielded by researchers worldwide. Their work not only earned them the Nobel Prize in Chemistry in 2020, but also set the stage for vast and varied applications, from correcting genetic defects to creating disease-resistant crops. CRISPR-Cas9's precision and efficiency have made it a cornerstone in laboratories focusing on genetic research and therapy. Its ability to target DNA with pinpoint accuracy means that diseases previously thought untouchable, such as certain cancers and genetic disorders, might one day be managed or even cured. However, with such transformative potential, the technology invites profound ethical considerations. As we gaze toward uncharted territories of human and ecological enhancement, we must ask, how do we navigate the potentials and perils of rewiring the very fabric of life? In the next chapter, we'll explore the real-world applications already unfolding and the ethical frameworks being developed to guide this extraordinary tool. While CRISPR's most celebrated contribution lies in its gene editing capabilities, a groundbreaking application is emerging in the realm of diagnostics. Researchers are harnessing CRISPR's precise targeting abilities to detect diseases with unprecedented accuracy. This innovation stems from CRISPR's natural role in prokaryotes, detecting and dismantling viral DNA. By repurposing this mechanism, scientists have developed CRISPR-based diagnostic tools capable of rapid and reliable detection of pathogens. One of the most significant advancements is the creation of CRISPR systems that can identify specific genetic sequences, making it possible to pinpoint diseases ranging from viral infections to genetic disorders. These diagnostics are not only fast, but also adaptable. They can be tailored to recognize emerging pathogens, which is crucial in managing public health crises like pandemics. The elegance of CRISPR diagnostics lies in its simplicity and efficiency. By delivering a signal upon detecting a target sequence, these tools provide a straightforward readout that can be interpreted without sophisticated laboratory equipment. This potential for on-site, point-of-care testing could revolutionize how we approach disease detection and management globally. But as we delve deeper into the capabilities of CRISPR, what ethical considerations arise from these powerful tools, and how will they shape our future? Enter CRISPR, a groundbreaking technology that harnesses the power of nature's own defense mechanisms. Originally discovered in prokaryotic organisms such as bacteria and archaea, CRISPR sequences serve as vigilant guardians against viral invaders. Imagine a microscopic memory bank where past encounters with viruses are recorded in the DNA, enabling a swift and effective response to future threats. Paired with the Cas9 enzyme, these sequences form a formidable duo capable of precise gene editing. This technology is transforming our approach to disease prevention. By identifying and neutralizing genetic threats before they manifest as illness, CRISPR holds promise for eradicating hereditary diseases and bolstering our defenses against viruses. From treating genetic disorders to developing innovative therapeutics, the potential applications seem boundless. The CRISPR-Cas9 system, honored with the Nobel Prize in Chemistry, is not just a tool for editing genes. It is a beacon of hope in the fight against diseases that have long plagued humanity. Yet as we stand on the brink of this new frontier, important questions arise. How do we navigate the ethical complexities of such powerful technology? And what responsibilities do we bear as we shape the genetic future of life on Earth? As we ponder these questions, we move closer to a world where genetic diseases may become relics of the past, sequences, a finding that became foundational for genome editing. CRISPR's ability to precisely target specific DNA sequences has opened vast new avenues in treating diseases. Using the CRISPR-Cas9 system, it's possible to locate and potentially correct genetic mutations, offering hope for conditions previously considered untreatable. This precision targeting isn't just a breakthrough in genetic editing, it's transformative for therapeutic applications. Inherited disorders like cystic fibrosis or muscular dystrophy could one day be addressed at the genetic level, directly repairing faulty genes. Moreover, CRISPR's potential extends into tackling complex diseases such as cancer, where targeting specific mutations could disrupt cancer cell growth without harming healthy cells. The system's adaptability stems from its natural origin as a defense mechanism against viruses, evolved to recognize and cut viral DNA with remarkable accuracy. This same principle now enables scientists to design CRISPR tools that can home in on any chosen genetic sequence. Yet, as we harness this power, ethical considerations loom. Who decides which genes to edit? And with precision targeting, what unforeseen consequences might emerge? As we probe these questions, we step into the next chapter, exploring the societal implications and the responsibility that accompanies this powerful tool. The discovery of diverse CRISPR-Cas systems in prokaryotes like bacteria and archaea has opened new avenues beyond gene editing, suggesting a vast, untapped potential waiting to be explored. These sequences, originally serving as a defense mechanism against bacteriophages, paint a picture of microbial adaptive immunity. Approximately 50% of sequenced bacterial genomes and nearly 90% of sequenced archaea harbor these CRISPR elements, showcasing their widespread evolutionary significance. Cas9, a pivotal enzyme in this realm, acts as a precise tool that leverages CRISPR sequences to seek and snip specific DNA strands. This has not only advanced our ability to edit genes, but has also fueled innovations across biological research, biotechnology, and medical treatments. Recognized with a Nobel Prize in Chemistry in 2020, the CRISPR-Cas9 technology represents a groundbreaking leap in our scientific capabilities. But our journey through the world of CRISPR is just beginning. As researchers uncover more about the myriad CRISPR-Cas systems beyond the well-known Cas9, there's a growing understanding that these systems might hold keys to new, unimagined possibilities. As we push these boundaries, we must consider what ethical frameworks will guide us as we harness this power further. This question leads us to consider the ethical dimensions and societal responsibilities that accompany such transformative tools. As we delve into the ethical considerations of CRISPR, the profound implications of manipulating life's code become clear. CRISPR, with its origins in the defense systems of simple prokaryotic organisms, now offers us the power to edit genomes with unprecedented precision through the CRISPR-Cas9 system. Yet, this power brings significant ethical challenges. The potential to correct genetic diseases is counterbalanced by concerns about unintended consequences and the moral implications of altering human germlines. The risk of off-target effects, where the technology might edit DNA at unintended locations, poses a technical challenge that scientists are still striving to overcome. Moreover, the prospect of designer babies raises questions about equity and access, as such advanced interventions might only be available to a privileged few, widening the gap between different societal groups. As CRISPR technology advances, regulatory frameworks struggle to keep pace, leaving a gap in governance that needs urgently to be addressed. How society chooses to navigate these dilemmas will shape the future landscape of genetic research and medicine. As we ponder these pressing issues, we must also consider the innovative potential of CRISPR to revolutionize agriculture and industry. How will this transformative technology redefine our understanding of the natural world and our place within it? Sequences by altering the guide RNA, a major leap that turned CRISPR-Cas9 into a precise gene editing tool. This breakthrough paved the way for CRISPR to evolve beyond its natural bacterial origins, becoming a cornerstone in modern genetic engineering. Today, researchers are exploring its potential to eradicate genetic disorders by correcting faulty genes, combating diseases like cystic fibrosis and muscular dystrophy. The medical implications are profound, yet CRISPR's influence might extend even further. In agriculture, it promises crops resistant to disease and pests, potentially alleviating food insecurity. With ongoing studies, CRISPR could enhance biotechnological innovations, resulting in eco-friendly materials and sustainable energy solutions. Yet, as we march into this genetic frontier, ethical considerations loom large. The prospect of editing human embryos or creating designer organisms raises questions about morality and long-term consequences. The evolution of CRISPR beckons a future where genetic interventions are as common as vaccinations, revolutionizing how we perceive life itself. As we stand on the precipice of this genetic revolution, we must ask ourselves, how will we navigate the power to alter life at its most fundamental level?